ways for us to understand the basic inheritance of discrete characteristics and discrete genotypes. So remember, and I know you've covered all of this before, so I know it's redundant, but bear with us. So what Mendel did, basically, took pea plants, and it worked well with peas, and it didn't work well with other plants, and there were reasons for that. But with his peas, he took purple flower peas, for instance, and white flower peas, and basically self-fertilized them over and over until he got lines of white fruit. So the whites always produce white, and the purple ones always produce purple. And then he crossed them with each other and discovered that in that, the next generation, which we call the F1 generation, so these are the parental population here, F1 here, and in the F1, he found that they were all purple. Okay? And then when he selfed his purple flowers, these purple flowering individuals, so one individual cell would produce offspring that were more or less 3-to-1 purple to white. And what he discovered by doing this, fortunately he was incredibly meticulous and kept incredibly good records, and he did this a bazillion times with a lot of different plant species and different characteristics, trying to figure out what was going on. But what these data show, the simple result right here, is that some of these organisms have some inherited thing, which we now call genes or alleles, which are different forms of genes, and they're passed on in a particular manner, which means, in other words, that that all of these individuals have particles, alleles that code for purple, and these have particles or alleles that code for white. And when you put them together, the purple ones, it only takes one copy of the purple ones, like a purple allele or particle, to cause them to be purple. And that's why all of these are purple, because they have one purple allele and they have one white allele. And when you cross them together, you take one of these and self it with itself, you wind up with 3 that have at least one purple gene and only one that has 2 of these white genes. And the reason for this is that the purple gene allows the organism to make a purple pigment in the flowers, and it doesn't matter whether you have 1 copy or 2 copies of it, you make the purple. So if you don't make it, if you don't have at least 1 copy, then you wind up making these unpigmented flowers. So these genes that are what we call dominant are expressed whether you have 1 or 2 copies. If they're recessive, they're only expressed if you have both copies. So white, you would have a white allele that's recessive and a purple allele that's not. So this brings us to some more good definitions. So we need to be able to distinguish genotypes from phenotypes. So what Mendel was doing is he was looking at the phenotype of the plant. So phenotypes are the physical manifestations of an organism that you can observe. They're physiological, morphological, biochemical, behavioral, and basically everything else about an organism that you can observe except for the genes themselves. You can now observe those individual genes, those individual codons or nucleotides, but we wouldn't consider them a phenotype, the pure genotype. And so the genotype are the genes, the genetic material possessed by an organism, a particular organism. And we might talk about the genotype referring to its entire genome, so the entire set of genes that it has. Or we might be talking, if we're talking about a particular trait or a set of individual traits, like flower color and pea color, for instance. And we'd be talking about the genotype just with respect to those particular genes, which importantly express particular loci. So when we talked about this the other day, loci are essentially positions on chromosomes for genes in growth. So what Mendel was able to figure out from data such as these, basically this proportion in the F2 generation and the proportion in the F1 generation, data that he was looking at. And he allowed us to figure out these two different laws, Mendel's two laws, the law of segregation and the law of independent sorting. And the law of segregation says that each individual has two copies of a particular gene, and that when gametes are made, those genes separate and they go to individual gametes. And that the law of independent assortment is simply that when you have multiple genes, they assort independently. So they don't, if you have a gene for flower color and a gene for seed color, those genes can go to the offspring independently. In other words, they don't, the purple and wrinkled, or wrinkled seeds don't always go together, and white and unwrinkled seeds don't always go together. It turns out that this isn't exactly true, because it's only true if they occur on different chromosomes. And it happened that Mendel was lucky to work with a bunch of traits that were on different chromosomes in these pea plants, and therefore they did assort nicely in these situations. There were other genes and other plant species where they didn't assort independently, and he didn't quite know what to make of that. But basically, the mathematics of the ones that did assort independently, because they were on different chromosomes, the mathematics were so striking that it made it very clear that this is what was going on. And so they basically, what this revealed is that organisms have, in fact, two copies of every gene, right? We now know that because we have two chromosomes, two copies of each of our chromosomes, and that that those chromosomes assort independently and take your genes with them. So Mendel's laws have become the basis for modern genetics with some caveats. But this plugged a really important hole in what Darwin was looking at, because Darwin and his contemporaries believed that organisms, that if you had two parents, their characteristics were simply blended and sort of mixed together. So you got an average phenotype when people had offspring. So it was some average of them. And the problem with this is that if you have rare but really beneficial traits, they can never express themselves enough to become selective. So it made it difficult for Darwin to figure out how certain characteristics could become